Good morning, friends. I'm sorry to interrupt your good conversation and getting acquainted with one another, but I'd like to start our program today. Please continue to enjoy your breakfast. We absolutely want you to enjoy that, and coffee on the table. My name's Lynette Marshall, and I'm delighted to welcome you here today. I have the privilege of serving as head of the University of Iowa Center for Advancement, and I can't think of a finer way to start a new week than really learning something and being able to come together and to hear from our colleagues in the Iowa Neuroscience Institute. Thanks for getting up early on a cold morning and finding your way here. We're delighted to be able to present 4 distinguished faculty members to you today. And as you know, they are all faculty from the Iowa Neuroscience Institute, and we're to be able to feature some of their remarkable research that's happening and the way they are working to advance figuring out some of these most difficult diseases that we deal with in our families and in our communities. I'll introduce the speakers in just a moment, but first I want to acknowledge that Troy Ross is here as Executive Director of the Roy J. Carver Trust, as well as his colleague Lynn Sazmajer. Thank you very much for joining us today. We appreciate that. You probably remember in 2016, the Roy Carver Trust made a breathtaking $45 million investment in this work that helped us establish the Iowa Neuroscience Institute. And that transformational support helped us recruit Ted Abel, who you will hear from first, and provided crucial research funding and support for faculty members. We're deeply grateful to the Carver Trust Board of Trustees, as well as Troy and Lynn, for your longstanding support. Thank you very, very much. Here today to tell you about the work of the Iowa Neuroscience Institute will be Ted Abel, who's director of the Neuroscience Institute, as well as interim chair of the Department of Pharmacology. Dr. Peggy Nopoulos, chair of the Department of Psychiatry and professor of Psychiatry, Neurology, and Pediatrics. Dr. Aaron Boes, director of the Noninvasive Brain Stimulation Clinical Program and assistant professor of Pediatrics, Neurology, and Psychiatry. And then Dr. Kumar Narayanan, associate director of the Iowa Neuroscience Institute and associate professor of Neurology. Our faculty members will be sharing with you how their work is changing our understanding of the neurodevelopmental, psychiatric, and neurodegenerative disorders. And at the end of the program, we'll have an opportunity for you to ask questions. So at the end, we'll bring all of the faculty up here, and you'll have an opportunity to ask them questions, and they can respond. So I'm going to begin by introducing Ted Abel. In addition to serving as director of the Iowa Neuroscience Institute, Ted is doing extra duty in serving as the interim chair of the Department of Pharmacology and professor of physiology and biophysics in the Roy J. and Lucille A. Carver College of Medicine. He holds secondary appointments in the departments of psychiatry, biochemistry, and psychological and brain sciences, and he holds the Roy J. Carver Chair in neuroscience. Dr. Abel has a distinguished academic career. He has an undergraduate degree from Swarthmore College, and he attended Cambridge University as a Marshall Scholar. He then moved to Harvard, and after receiving his PhD in biochemistry and molecular biology, he completed his postdoctoral work in the College of Physicians and Surgeons at the Columbia University. Today, Dr. Abel's research focuses on the molecular mechanisms of memory storage and the molecular basis for neurodevelopmental and psychiatric disorders. He's been a pioneer in the use of molecular and genetic approaches to define how neural circuits mediate behavior, including identifying the molecular impact of sleep deprivation. And I thank someone for the notes. Ted, we're delighted to have you here today. I know they're going to enjoy hearing from you. Please join me in welcoming Dr. Ted Abel.
Are we going to switch it here? And I promise that there won't be any tests, unlike after many of my lectures. Are we ready to go? Okay, great. Let me echo Lynette's welcome this morning. Thank you very much for taking time out to join us for breakfast and some discussion about neuroscience and how it impacts all of us in our families and our friends, and how we can begin to identify and develop novel therapeutic approaches. So psychiatric and neurological disorders are devastating. They're devastating to the patients who suffer from them. They're devastating to the families that have to support those patients. And they're devastating in terms of their financial and societal impact. Our goal in the Iowa Neuroscience Institute is to undertake fundamental research in neuroscience to try to better understand these conditions and eventually develop better therapeutic approaches to improve the lives of those who suffer with these disorders. You'll hopefully hear some of that today, see some patients who do better in many ways thanks to the therapies that we have, but the therapy remains, as you know, a major challenge. The brain is really one of the complexities of nature that we really understand very little about. We have a lot of facts, I would say, but very little understanding. The human brain is 3 pounds of tissue. It contains 86 billion neurons. Each of these neurons makes 10,000 connections with other neurons next to it or across the brain from it. And together, all of these give rise to our memories, our emotions, and our thoughts. And that simply is our challenge. It's a challenge that we're not going to succeed at. We may succeed at pieces of it, at providing some insights and hopefully some therapies. It's obviously been of interest to artists like Salvador Dali and writers like Marcel Proust, and even the little less intellectual Inside Out with Pixar, with its demonstration of memory as a sort of balls on a shelf, and fear and anger as locations in the brain. I should say one of the hallmarks now we know of the nervous system is that there are these circuits of neurons that are located in particular parts of the brain that mediate different behaviors. That's kind of what's being drawn here. And what that view of the brain, that there are parts of the brain that mediate memory and parts that mediate fear and parts that mediate positive emotion, derives in large part from work carried out here at the University of Iowa by the Damasios, who started studying patients with lesions, neurological patients that had damage, often due to stroke, to particular brain regions. And the Damasios and now researchers here who have followed them use those patients to identify the function of particular brain regions. One striking example was a patient, SM, who had a lesion in her brain, a region called the amygdala, because of a genetic disorder. And that led to a dramatic demonstration that the amygdala was critical for our ability to sense fear and be afraid. And that work came from the University of Iowa hospitals and clinics here. Now, what we're interested in is how these behaviors emerge from these neural circuits, and we have a number of modern tools to carry out these studies. A less modern tool is functional MRI imaging that many of you know about, where you can literally image the action of neurons in the brain. Here's neurons in the brain region called the striatum that respond to reward. We can use modern cell and molecular-based techniques to fluorescently label cells. Here's an approach affectionately referred to as rainbow, where you can make neurons have a rainbow of colors and study what they do. In a more biophysical way, you can record electrical activity from large numbers of neurons simultaneously. One of our new hires is able to do this in the mouse in 7 different brain regions. Our neurosurgeon colleagues do this in patients with epilepsy by putting a mesh of electrodes over a temporal lobe of a patient to identify where they are having seizures. And some of our researchers use a technique called optogenetics. Here is a schematic from a playwright called Tom Stoppard, who you may know from writing Rosencrantz and Guildenstern Are Dead. One of his more recent plays is called The Hard Problem, which is literally about what we are trying to study. And in this play, which is about 5 years old now, he talks about the use of optogenetics, which is lasers, to control the function of neurons. One of our new hires in pharmacology and in the Neuroscience Institute studies circuits that drive feeding in a brain region called the hypothalamus. And he's able to engineer mice such that when you shine light on neurons in the hypothalamus, the animals stop whatever they're doing, or they wake up if they're asleep, and they go and eat. I've urged him to develop the opposite, to get them to stop eating, but it turns out it's easier to get them to eat, as may resonate with many of us. So there are some striking experiments. My own work in my lab focuses on the persistence of memory, trying to understand how it is that a single event, we do experiments in mice that last 3 minutes long, and they can remember that event for at least 6 months. We've done experiments for sort of that long. We haven't gone any longer. My students wouldn't have it if we had to wait longer than 6 months. What I've discovered as I give these more general talks is that Salvador Dali has really portrayed a lot of what I study. The persistence of memory here with his clocks and driving memory, the decline of the persistence of memory with memory fragmenting. And in the middle, his painting Sleep. One of the large parts of my lab looks at the role and the way in which sleep can drive memory storage. We've been able to identify some of the molecular processes that are literally replayed during sleep to enable us to store memories. And then lastly, a part of my lab now focuses on neurodevelopmental disorders, but we want to try to understand how it is that all of our knowledge of basic neuroscience and our fundamental knowledge of the molecular mechanisms of memory can help inform us about how cognitive deficits can be improved in patients with autism, with schizophrenia, with Alzheimer's disease, with Parkinson's disease. Now, we're obviously interested in a number of these diseases, from autism to Parkinson's to Alzheimer's, and one of our goals now in the Neuroscience Institute, we've been kind of getting off the ground for the past 3 years with 12 new hires and over 100 faculty members across the university. And we're beginning to identify clusters, you'll see some of them today, where we have a group of researchers with expertise that connect to the clinic. We hope eventually to be able to develop centers dedicated to these various disorders. I know firsthand the cost of brain disorders. Our son Seamus, here is a picture from about 10 years ago, is on the autism spectrum. He's very high functioning, but suffers from autism. He's now 19 years old, just turned 19. And he's never had a friend. His best friend is his grandmother, and he's an only child but really it's pretty amazing to help him and really learn how he can navigate through life. So the movies you see and the stories you see of kids who walk into a shopping mall and then run out screaming because they can't deal with the music that's being played, that's something that my wife Noreen and I and my mom, she goes by Annie, that we've experienced, very often in our lives. The challenge right now is to help Seamus not navigate school, but navigate life. And what I think everyone doesn't realize is that the kids with autism grow up to become men and women with autism. And finding a way for them to make their way in the world is a major challenge. And we're in the process of that. I was reading chapter 1 of Jane Eyre with him over the weekend, and he has to have it as audio as well as reading. And then, is it John Reed, I think the guy in there that is kind of a bully, and Seamus all of a sudden starts crying because the Jane Eyre in this case is being bullied by I guess it's her cousin, maybe John. And so it's weird to see these interactions and how they someone who really doesn't seem to have emotion and connect to people in life, he actually has much more reactions to things that he reads. So these are devastating disorders, and it's a daily challenge for many of us to navigate them. So to understand and treat these disorders, we've developed an interdisciplinary group of faculty. Here is a picture of one of our speakers today, Kumar Narayanan, very striking picture with Nancy Andreasen, a senior thought leader in the field of psychiatry who won the National Medal of Science, Crystal Parker, who was a trainee with Nancy and Kumar, and the first hire in the Neuroscience Institute, a woman who received an NIH R01 grant on first application. So NIH grants are funded at about 10 to 15%, so that is quite an achievement. And this illustrates some of the variety of techniques, many of which I've had a chance to touch on, that we're using. And we have faculty that range from neurology and psychiatry to engineering and also education. We've developed a number of educational initiatives. One of them is an undergraduate major in neuroscience. that is about a year and a half old and enrolls 185 students. Our PhD and MD PhD program is 30 years old. It's really an impressive PhD program. And we have a large number of medical students that participate in research, as well as a postdoctoral fellowship program. This one, called Inspire, is led by Peggy, who you'll have a chance to hear from. We've even gotten Herky in the mix here. I couldn't resist taking this photo at one of the undergraduate teaching labs. I'm not sure Herky can even see anything. But the whole lab coat thing was pretty dramatic image. So, yeah. And lastly, really, to do all of this, we need extraordinary resources. Our most extraordinary resource are our people, and they are people who know how to party, actually. This is a party at the Society for Neuroscience, mainly grad students and postdocs with the odd professor, Crystal and John. Peg's there. Yeah, so yeah, you are. So we had a few of the survivors. When I was at the University of Cambridge, they have these things called May balls, which are start at dinner and end at breakfast, and they take a survivor's photo after breakfast. Yeah, I have these survivors' photos on my wall. So this is sort of the survivor's photo of one of the Iowa Neuroscience parties. Extraordinary buildings built by someone in the room. So thank you very much, Steve, for building PVDB. And then extraordinary financial resources from all of you who contribute to us, and particularly Troy and Lynn. Thank you so much for the gift from the Carver Charitable Trust. I want to end with a quote that those of you that have heard me speak before, I always say this, and I think it's important to keep thinking about it. And it's a quote from Francis Crick. Francis Crick, along with Jim Watson, was a co-discoverer of the double helix of DNA. I should say he was a triple discoverer along with Rosalind Franklin, who actually did the experiments that they used to build their model. And I think what Francis said, by the way, I should say about Francis made dramatic contributions to structural biology, and we know that work from the double helix of DNA. He made dramatic contributions to what we call the central dogma of molecular biology, which is DNA makes RNA makes protein, studying the way RNA is turned into protein. He then turned to neuroscience. He did not make stunning contributions to neuroscience. He had some very speculative papers, but it's worth noting that a very, very smart man who could have won 2 Nobel Prizes but only won 1, ran into neuroscience and hit an intellectual wall. Okay, so at least realize what we're up against. Okay, and what Francis said is that it's true by blundering about we stumbled on gold, but the fact remains we were looking for gold. An appropriate Iowa quote, it turns out, and I think it also underscores the fact that what we do in the lab is not what we did when we were in intro chemistry, where you write a purpose and then you have little results and you measure everything and you end with you're well within 3% of what we expected to see, because we don't know what we're expecting to see. We're hoping that we find new things, and our sort of job is to set ourselves up for discovery and to stumble a bit, and that's where we need all of the help of those resources from donors and from the foundation that enable us to do that. So let me finish there. Hopefully I'm not too far over time. We're pretty good. We're in pretty good shape. I think what we'll do is have a number of the talks, and then we'll have a discussion session afterwards, if that's all right. And let me get Peg's talk up and introduce her. All right. So our next speaker is Peg Nopoulos. Peg is an Iowa native. She did all of her training here at the University of Iowa as an undergraduate and then as an MD PhD student, and has risen through the ranks, very much risen through the ranks, cracking various glass ceilings as she goes, which is tremendous, and is now chair of the Department of Psychiatry. Peg is really one of the very few women leaders in the field of psychiatry, and it's great that she grew up here and is here to lead us. Peg, we're looking forward to hearing from you. Peg will tell us about her work on neurodevelopment and psychiatric disorders, and it should be an entertaining talk.
So,
Thanks, Peg.
I am an Iowan, And yes, that means I'm Iowa nice. So just a couple clarifiers. I actually don't have my PhD, MD only. No. And no, I don't need an honorary PhD. And it's just, so the fact that I'm chair in a department that I grew up in is really quite humbling. Because I'm now, directing some of the people, most of the people who trained me. And mostly on a daily basis, I forget I'm chair, which is actually probably an okay thing. Because it's just a wonderful place. I feel like, much of my success has been the environment in which I've been able to work, which has really been nothing short of phenomenal. So we're going to talk about neurodevelopment, kind of a 30,000-foot view of things. So initially, just a primer on the brain to make sure that everyone's on the same page. And then human brain development. And if you learn one thing from this talk, is that it takes a long time to fully develop a human brain. Okay? That's one thing you walk away from. Long time. And then how learning about development can help us understand neuropsychiatric disorders. So a little bit on the primer. So you're going to look at MRI scans in particular. Just want you to orient how you would look at this. But these are some of the terms. Cerebral cortex, which means bark, actually. So it's like the bark of a tree. Some of the internal structures, the cerebellum. But looking at a microscopic level, it's really important to think about how we name these things. So a brain cell is called a neuron. The dendrites, okay, these are what are the branches, and these are what communicate with other neurons. And remember that neurons don't work in isolation. They work with billions and billions of connections. And that's how they make those connections, is by the dendrites. The cell body, which is where all of the functions happen, all of the metabolism that happens in the cell. And then the axon, which is sometimes a very, very long process, which also reaches out to other cells. It also sends electrical impulses. So this is where what we call myelin covers this. And myelin is like the cord or the wrapping of an electrical cord. So it allows the electricity to go faster. It's an electrical cable in many ways. And then the all-important synapse. So the synapse is where the cells exchange chemicals. So once again, everything about the biology of the brain really comes down to electricity and chemicals. But that's how the brain functions. Electrical impulses that release chemicals, and cells talk to each other. So here's this function again. Let me look at it much more closely to kind of give you a sense of how these cells are laid out in the human cortex. So those are those dendrites that I'd mentioned. And then the cell bodies and another cell layer. And once again, the cortex is divided into these layers, and that's the axons. And that makes up the gray matter, which is the cortex, which is made up of the dendrites and the cell bodies. And the white matter, those cables, all of those cables, makes up the white matter. And those are the axons. And so this is the MRI scan that I told you about, and that's the gray matter or the cortex, that's the white matter, and then the black spaces are the cerebral spinal fluid. That's the fluid that surrounds the brain. So these are the basic tissues that I'm going to talk about in terms of how things change over time in the brain. So the important thing, and Ted mentioned this, is that the brain regions work together. We spent a good 100 years figuring out that this part of the brain works different than this part of the brain. This part of the brain does something different. But really, in the past 10, 20 years, some of the most important findings are the fact that there are circuits. And each circuit has a specific function. It's a little bit of an oversimplification, but this is a picture of those axons. So these are the highways, and these are the ways in which regions of the brain talk to each other. And this is just a schematic to kind of give you an idea of the fact that there are regions of the brain that are constantly talking to each other, and each circuit has a different function: mood, memory, attention, reasoning, social behavior. So how about development? How do these circuits and the brain development over time? I'm going to talk about very basic MRI, which is structural imaging. Ted mentioned the functional imaging, where you actually can look at functional connectivity, or looking at cells that are actually working. This is just looking simply at quantitative measures, volume of gray matter, volume of white matter. Really basic stuff, but important to know, because that's some of the dramatic things that change over time. So here's some of the most important changes, some of the most dramatic changes, and you might expect this. It happens from birth to 1 year. So these are MRI scans of babies who are just born and 1 year later. And you can see here, this is the volume. And you can see that the volume increases really dramatically in that first year, over 100% of volume gain. And then you can actually see that even though there's a little bit of increase between 1 and 2 years, it's not as dramatic. This first year of life, the brain really expands greatly. It's a huge amount of development. So that's a really important time, that first year. And then on to the school age sample. I will tell you that one of the reasons why we don't know an awful lot between age 2 and 6 is because it's hard to image those kids. Because they move. But we have a lot of work on school age, so starting at about 5 or 6 on up. And here's something relatively striking. By the age of 6 years of age, the total volume of the brain is about 95%. So a 6-year-old, okay, this is the volume of total brain from 5 to 25. It doesn't change much, okay? That's a little striking. It doesn't seem to change much. However, even though the volume doesn't change, there's a dramatic change in tissue remodeling. So even if the volume doesn't change, how the gray and the white matter changes over time is really what's happening. So an analogy is the tree in the brain. So remember, here's the neuron that I told you about. That's the cell body, the axon. If you can think about the dendrites as branches and the trunk as the axon with roots. And one of the things I'm going to talk about is pruning. And the reason we think about that as a tree or a bush is because pruning is actually a good thing. So starting around the beginning of adolescence, we see a pruning of connections. So we're not losing cells, okay? We're not losing trees. We're just losing connections. So here's our fancy little neuron, and then you can see that there's pruning. And once again, this is a normal developmental phenomenon, and it's a good thing, right? Because what happens when you prune your bushes? They grow better, right? So you're kicking off those dendrites that are not important, and you're strengthening the ones that are there. So pruning is a good thing, but it leads to a dramatic decrease in the volume of the cerebral cortex, just losing those connections. Every time I see this, it totally freaks me out because this is your brain on adolescence, okay? This is cerebral gray matter volume. These are normal, healthy children. And this is 5 to 25. This is a normal process, okay? And that's the pruning. Remember, you're not losing cells, you're just losing connections. But it's really important to realize that there's a really big conundrum between what we call mature, right, 18, by societal definition, versus biologically what really happens in the brain. So this is what I call the slippery slope of adolescence. So just to give it some context, the 12-year-old, the 14-year-old, we give them a driver's permit. Here, take the keys. Okay, you're losing gray matter faster than anything else. Here, take the keys. Go ahead, just start driving, okay? And then by 16, we're, okay, go ahead, take the keys, and now you can drive alone. You don't even need any supervision, just go out there. By 18, once again, still on the slippery slope, but by that time, go ahead and vote. You go to the military service, right? These are all things that we consider to be societally defined by what maturation is. And then at age 21, go ahead and drink. I think the issue is, once again, even though what we call societally maturation is not biologically what's happening in the brain. And once again, if you don't take anything from this, by the way, those are my kids. Oh, what a coincidence. They're a little bit older now, but they're all still in that, what I thought, toddlerhood was a problem, but the years between 18 and 25, oh my goodness. But, and this actually stops at 25. Others have done studies, and we've recently done some studies, where some of these changes really continue through age 30. Okay, so once again, it takes a very long time for brain maturation. This process is later in life, obviously beyond what you might consider developmental windows. But once again, because there's a tissue remodeling, so you're losing gray matter, but at the same time, you're gaining white matter. So remember, white matter is those roots, and it's the cables that connect neurons to each other. And so as you're losing the gray matter, you're building the white matter. So there's tissue remodeling, so that age between 5 and 30, even though the volume isn't changing a lot, what's happening inside the tissues is dramatic and dynamic. So the changes are mostly in tissue distribution rather than volume. They're robust changes. These are not minor changes. And it could be termed late maturation rather than development. It's all kind of terminology. And these changes occur over a prolonged period of time. It takes a long time to develop the human brain. So how about learning psychiatric disorders in the context of this development? I think, although it might be, so this is a little heresy, a little oversimplification, I think everything begins in abnormal development. And let's take a little walk through that. The factors that create different disorders, even though everything is based on abnormal development, can be boiled down to how severe the developmental problem is, and also can the rest of the brain compensate for a badly developed circuit. So let's just do a little walk through possibilities. First year of life, remember that's one of the most important changes in some of the early development. And in general, I'm going to say genes affecting brain development, but of course environment impacts on this too. I don't want to be overly simplistic, but a lot of the determinants of what we know affect the development of psychiatric disorders and neurologic disorders have strong genetic components. So the genes affecting brain development, if it's severe enough, right? So here's the key. If it's a big arrow, so it's a severe abnormal development, you're going to manifest disease early, okay? So the brain can't compensate because that circuit that's developed is really abnormal, or it's several circuits. And you're going to get infant diagnoses. You're going to get intellectual disability. You're going to get autism, okay? If you move on to abnormalities that are a little bit more moderate, once again, driven by genes, probably in particular a particular circuit. If your brain can compensate through infancy, this disorder may manifest in childhood, such as ADHD. There's a lot of these that manifest in childhood. If we walk through this further, so once again, an abnormality in brain development, but now probably less severe, because the brain can compensate through childhood. But these are disorders that develop in adulthood: addiction, depression, schizophrenia. Those maturational changes, so if you have an abnormal circuit, if you developed an abnormal circuit but could compensate that, once you go through that pruning process, whoa, that may very well uncover the abnormality in a circuit. Now we go on to something that's even more heresy. Even neurodegenerative disorders are caused by abnormal brain development. Woo, there it is. What do you think, Kumar?
Not a mild problem.
It's not a mild problem, but the mild means the developmental aberration. So how abnormal is the development, right? If it's a severe abnormal, you're going to manifest early. These are diseases that happen later in life. But let me just tell you briefly about the disease I work on, which is Huntington's disease. It's a single gene disorder. It's a disorder that we can look at lifelong. I can look at children who have the Huntington gene Children who have the Huntington gene are going to develop Huntington's when they're 50 years old. But guess what happens when they're 6 years of age? Their brain is already abnormal. And in particular, in the circuit, it's a very specific circuit in the striatum, the basal ganglia. As early as we can see, those kids that are going to have a degenerative disorder have abnormal brain development. Why don't they manifest it when they're kids? Because their brain can compensate. So the developmental problem is compensated for. Another part of the brain comes and helps out, so that later, it's only later on in life when essentially that compensation fails that then manifests. So this is not to say, of course, that these are minor problems. I'm just talking about the severity of the developmental problem. So this is an important reason why we can think about, a little bit of work in Alzheimer's disease. There's one gene that we know is important for developing, so ApoE. We know that ApoE is something that in youth actually confers an advantage in some things. Same thing with the Huntington's gene. It's actually a gene that helps the brain. So early in life, it can be helpful, gives an advantage in cognition. But later in life, causes the degeneration. So this is an overview of this concept of how everything, so Freud was right, everything starts in the beginning, in the context of development. So on to this, now I'm going to tug on your heartstrings. I'm going to talk about, in general, how people with mental illness enrich our lives. And some of this has to do with the fact that I'm going to show you pictures of people that you know that suffer from mental illness, but what's really important is that mental health and disorders of mental health are the most common scourge of humankind. But they're still shrouded in stigma. Let me just do an emotional experiment with you. What do you feel when I say the word soldier? Versus what do you feel when I say the word addict? So in the Vietnam War, we shut down campuses. Young men are dying, young men are dying. In 2017, we had more people die of heroin overdose than the entire Vietnam War. What's up with that? It's because of that feeling experiment, right? Addict. That's not my son. That's not my dad. There's still a huge stigma surrounding mental illness for no better reason, for the most part, than ignorance. Some of it is because the brain and those parts that make us most human, that are the root of mental disorders, is still an enigma. But we're learning. So here's the little tug on your heartstrings. gets me every time. Once again, those are people we know. But each one of us could play that song with members of our own family, right? But that's where we're at in the context of learning more about brain and behavior. So, actually, now I'm going to introduce Aaron Boes. It's my pleasure. Because Aaron started out under my tutelage. So because I'm an MD only, not a PhD, it was later in my career that I actually started hosting PhD students. Aaron was my first PhD student, MD PhD. So he's what I call the Green Berets, MD PhDs, like Kumar, know what they want. How do they do that? Know what they want so early in life. So they go to medical school, then they come into a lab, do a PhD, go back to medical school, and that's what Aaron did. Aaron came to my lab, and because he was a first PhD student, I didn't know what to expect. But he was awesome. He was a star. He was able to write papers that required little revision by me, which is totally unheard of. Then he left to become a pediatric neurologist. He did an internship in San Diego and then training at Harvard. And after his training as a pediatric neurologist, because of some of his work here with the Damasio work that Ted had talked about in terms of looking at lesions and circuitry, he wanted to learn more about transcranial magnetic stimulation, and he's going to talk about that. But it's a phenomenal tool that we use to treat and to query, to learn about brain circuitry. So he stayed and did a fellowship in transcranial magnetic stimulation. And so here's a pediatric neurologist who is now trained in a treatment that we use for adults. So I had to convince the chair of pediatrics that here, this young man coming into pediatrics is actually going to open up a TMS center and treat adults with mental disorders. I'm like, it's going to be a great thing. And it was. We did not have a TMS center until Aaron came. And I knew once he left my lab that I was going to recruit him back. I did all I could. I was so happy that we were able to do that. And he came at a phenomenal time right after Ted got here and was really able to be integrated in some of the things that Ted's been building. And so it's been an awesome last several years. And Aaron's going to talk to you about his work in TMS.
All right, thank you very much. So my name is Aaron Boes. I'm from Emmitsburg, Iowa, up in the northwest corner. And I get to talk to you today about one of my favorite topics, which is brain stimulation as a therapy. So I think probably everybody would agree with me that this last 100 years has really been a golden age for pharmacotherapy, or using medications to treat brain disorders. I think if you look at where we're at right now relative to where we were 100 years ago, there's been a really huge transformation societally where people who had disorders, either neurological disorders or psychiatric disorders, were really often on the fringes of society. They were sometimes institutionalized, they were sometimes homebound, and I think that's often not the case now. So we've made major inroads into treating disorders of the brain. Maybe not.
I think it is.
Peggy might have blown this.
Yeah? Can you turn it up a little bit? Yeah. Is that any better? Can you hear me in the back? Yeah. Okay. So I was just saying that we've made major inroads in terms of using medications to treat brain disorders, but we haven't made it all the way to where we want to be for sure. So there's still a lot of room to go. I think if you look across most brain disorders, we're able to treat about 2/3 of patients, with about 1/3 that really don't respond really well to medications. And that's across disorders, whether you're talking about epilepsy or schizophrenia. There's really very few disorders where we're at a full 100% treatment rate. And even those where we are able to treat, often we're not able to treat as effectively as we'd like. So it's very rare where we have actual medications that resemble cures where people are fully functional and back to their normal self. So it's often just taking care of symptoms, improving symptoms, allowing people to live better lives, but leaving a lot of room for improvement. So of those treatments that are non-pharmacotherapy, some of the very best ones that we have are actually using electricity to treat the brain. And I think that that is a huge potential that's been relatively unexplored in the field of neurology and psychiatry. So look at something like ECT, which is really one of the very best treatment modalities that we have for treating things like severe depression. If you have suicidal thoughts, ECT is often something that's able to dramatically improve your mood, your level of depression, in the order of hours, days. And there really are very few medications, or maybe even you could argue no medications, that are able to have that dramatic of an effect as ECT. But it's not without its side effects. So ECT does have, in some instances, cognitive side effects, memory side effects. So there's certainly room for improvement there, but ECT is highly effective. And then right next to that is deep brain stimulation. So that's slightly more sophisticated, relatively newer, developed in the 1990s, where you can actually implant an electrode deep inside somebody's brain and stimulate very focal targets, subcortical circuitry. And that's very, very effective for things like Parkinson's disease, dystonia, essential tremor. So across those disorders, it's often one of the most effective treatments, but very few indications in terms of, if you think about the overall number of disorders that we have across neurology, psychiatry, things that could potentially be effective for. We're really only scratching the surface, I think, with those few disorders that we're using it for. And then if you look on the far left here, these newer modalities, so I think these are areas where we're trying to essentially mimic what's happening with more invasive stimulation, whether it's ECT or deep brain stimulation, but in a way that doesn't require surgery, that would be much more available to the broader population. So people who maybe aren't depressed enough to require electroconvulsive therapy, maybe they aren't in a situation where neurosurgery would make sense, using something like deep brain stimulation, but something where we can augment what we're doing with medications with things like TMS, transcranial magnetic stimulation, which I'll talk more about, or transcranial current stimulation. So TMS, or transcranial magnetic stimulation, is an area of neuromodulation or brain stimulation that's very near and dear to my heart. And the idea is essentially that you just send electric current around copper wires that are embedded with plastic, so you can see that there. Creates a magnetic field. If there are any physicists in the room, that fluctuating magnetic field will actually stimulate the brain electrically. So it's a way to, you could think of it as sort of injecting an electric current in a focal area in the brain, and we can use that to modulate structures or change their level of excitability. So this is actually not that new of a technology. I think we often think of it as new now because we're using it in new and innovative ways, but it was actually developed all the way back in 1984 by Anthony Barker. So this is a British scientist who's still around. I actually saw him last week at Vancouver at a conference. So 35 years old is what that makes TMS. So it's been around for a long time. And you can see that he's stimulating a motor area in the brain. And if you look here, this hand is hooked up. It's got EMG equipment. So it's going to be recording movements of the hand. And early on, TMS was essentially used to measure excitability of the brain using this motor system as a metric. So stimulate the motor cortex here. You can measure what happens here. And as a research tool, you can start to understand what sorts of things change brain excitability. You know, as caffeine, medications, sleep deprivations, what kinds of those things will change the excitability of your brain. And then if you jump forward 10, 20 years, we started using TMS in a different way because new technology was developed that allows you to deliver it rapidly. So you can deliver rapid pulses of TMS, and that changes the excitability for longer periods of time. So you can do a 20-minute stimulation session, but have excitability changes that outlast that. And if you do it daily, you can actually have much more durable changes. So this was a mentor of mine when I was in Boston, so Alvaro Pasqualeone, and he's accredited to be the first one to use TMS as a treatment for depression, and that's something that's still being used today very frequently. But the idea is there's this area in the left prefrontal cortex, the left frontal lobe, that tends to be underactive in people who have major depression, and this is a form of brain stimulation that excites that area, or it makes it more excitable. So if you deliver it daily over a course of about 4 to 6 weeks, about a little more than half of people will have a really nice response to that, even after they've failed multiple medications. So somebody who's failed as many as 5, 6, 10 medications may still respond very well to this. And this is just a modern example of what this looks like today. So we have Meg Venture equipment here at the University of Iowa, and people are generally referred to us after they've failed somewhere in the range of 5 to 10 different medication trials. So for whatever reason, that's an individual who fits into that category of a third of individuals who are just really not responding well to medications. So they're referred to us, and if they're interested, we offer them this treatment with transcranial magnetic stimulation, which takes somewhere in the range of 3 to 37 minutes for a treatment session, so depending on which protocol they choose, and lasts about 4 to 6 weeks. For whatever reason, it takes about two weeks maybe for it to really have an effect. And like I said earlier, a little more than half of people will have a really great response to this. About a third of people will go into full remission from their depression from this. So often these are people who have, spent the last 10 years severely depressed and have almost forgotten what it's like to not be depressed anymore. So it varies in terms of how effective it is. There's room for improvement, and that's something that we're trying to do research on to understand why it works for some people and not other people, and how do we make it more effective. But for those who do respond, it can be transformative for them. So this is sort of my vision. My ambition in my career is to develop a University of Iowa Center for Non-Invasive Brain Stimulation, and I think we have all the components here to make a really world-class contribution to this field. So I think we're well situated to deliver outstanding patient care. I think that's something that we're already doing, staying at the forefront of what's available TMS-wise. Research, I think we're also well positioned because we have a lot of great collaborations across disciplines, which is absolutely essential for this field. So it's very technology driven, so you need a lot of engineers involved, you need a lot of statisticians involved, you need a lot of clinicians involved. So ultimately, the most effective research centers are going to be those that have the best collaborations, and I think we're doing an outstanding job of that so far at Iowa. I'll mention just one example, something that I'm probably most excited about in my research portfolio now, which is collaboration with neurosurgery. So TMS, while I've mentioned that it's been around for 30 years, 35 years, it's still largely unknown how it works, right? So a pretty basic question that, if understood, might be able to allow us to treat a lot more brain disorders besides just depression. So the thing that's challenging is you're doing TMS, and the brain is essentially in a black box, right? So it's in your skull. There's no way to know exactly what's happening in real time in terms of how you're changing neurons, and especially when you're stimulating an area that doesn't have a direct readout like the motor cortex where you can measure movements. So to circumvent that sort of black box challenge, we're collaborating with neurosurgery, and we're the only group in the world that's doing TMS in neurosurgical patients that have intracranial electrodes where you're monitoring brain activity in real time. So it's a really exciting area, so we have a window into the real-time activity changes that happen with TMS, and I think that'll be really informative in terms of how does TMS actually work, what can we do to change it, to make it better, to treat more disorders. So I think that's one of the areas that's exciting that was only possible at a place like this where people are highly collaborative and interested in working together. And then, of course, education, always one of the major missions at the University of Iowa. And I think we're well positioned for that as well across disciplines, psychiatry, neurology. We're training a fellow right now who's interested in neuromodulation and carrying forward the field. So with that, I just want to thank the Neuroscience Institute and the Carver Trust that's been hugely supportive of our work thus far in terms of providing equipment and providing resources to help get this program launched. And I look forward to any questions later. And then I also want to introduce Kumar, who's going to be talking about neurodegeneration, and hopefully also mention deep brain stimulation, which is something he's doing as well. Thanks.
First of all, thank you so much for coming in on this warm March morning to listen to our the exciting things going on in the Neuroscience Institute here at the University of Iowa. My name is Kumar Narayanan. I'm in the Department of Neurology, and I'm also in the Neuroscience Institute. Wow, this is exciting. So I wanted to start off by thanking the Carver Trust for their incredible support of our efforts. We have a research program of excellence. They've also provided pilot funding. They've been enormously supportive, so thank you. And hopefully you can see where some of those investments have gone. So I am a neurologist, and I specialize in Parkinson's disease. Every Tuesday morning, I see 7 or 8 patients with Parkinson's disease. And let me tell you, it's devastating. I have to tell these patients week after week that they're going to get worse every single day of their lives. I can treat their symptoms, but I cannot change their overall disease trajectory. And you might think, well, this is, patients just at the University of Iowa. This is something that affects a limited population of people. But when I think about my experience, I don't think about a neurology clinic, a hospital, I think about a tidal wave. And the reason I think about a tidal wave, see there's Parkinson's disease. I also see other diseases like this, Alzheimer's disease, other dementia and cognitive disorders. So the primary risk factor for Parkinson's disease is age. As you live longer, your chances of getting diseases like Parkinson's disease, Alzheimer's disease, and dementia and cognitive disorders go up. And as we've made enormous advances in medicine, treating cardiovascular disease, living longer, living healthier, we are going to get diseases like Parkinson's disease, Alzheimer's disease, and dementia in greater and greater quantities. And these diseases really rob us of our humanity. They take away every aspect of what it means to be human. And so we're looking at a society by 2040 where patients have these diseases in enormous numbers. And it's 2019. We have all kinds of amazing technologies. We have iPhones, we have apps, we have self-driving cars, self-driving tractors. Some of my patients who have Parkinson's disease who can no longer go out in the field driving their tractors with their phones. But we have no disease-modifying therapies for Parkinson's disease, for Alzheimer's disease, and dementia. We have nothing that changes the overall disease trajectory. And so this seems like a big problem, and you might think, well, there are a bunch of drug companies who are working on this. They're going to solve this problem, right? No. A lot of these companies looked at these diseases and they thought, wow, there's no way we're going to be able to make money off our investments in these diseases, and they pulled their investments. This is not just true of Pfizer, it's true of Merck, it's true of basically most major pharmaceutical companies. So it's up to us here in academic situations where we can try to figure out new mechanisms for the diseases to make a difference. And so, you know, our interest in dementia, our interest in Parkinson's disease, our interest in Alzheimer's disease, if we can convert that into basic science insights, into breakthroughs, we might be able to motivate these companies to really make the investment it takes to get a drug to market. So I'm going to tell you about 3 things that are going on in my lab. With every neurodegenerative disease, there are sort of 3 basic problems. The first is you want to diagnose these patients as early as possible. Because if there was a therapy that was going to protect failing circuits, you'd want to give it to patients as early as possible. The second is we want to know what are the basic mechanisms at work. What changes in Parkinson's disease? What changes in Alzheimer's disease? How can we make a difference as early as possible? And the third thing is we want to know if we know if we know the change with neurodegeneration, can we fix these failing circuits? And you might think that this is impossible. How do you fix a failing circuit? But I'm going to show you an example of how it works, how we use it every day in clinic, and how we might use it in the future. So with early diagnosis, one thing about the brain, all of our brains are highly dynamic. And if we can figure out how to capture that complexity as early as possible, we might be able to figure out, you know, this patient has Parkinson's disease, this patient is going to get Alzheimer's disease, this patient is going to get Lewy body dementia, and we might be able to do it very early. So one free way to look at brain activity in, you know, awake patients without going into the brain is an old technique. It's almost 100 years old, 99 years old. It's called EEG, electroencephalography. It's been used by neurologists for seizures, but we're finding that we can put these electrodes on patients, on the scalps of patients with Parkinson's disease. It has no side effects other than messing up your hair. And by using, and this is one of the most unique things if you're about the University of Iowa, we have world-class medicine, but we also have a world-class research institution. The University of Iowa has undergraduate departments across the river. And so when I showed one of these groups in electrical and computer engineering, these EEG signals, they immediately came up with a way to use all kinds of high-falutin math, which I'm not going to go into, but it's basically speech processing. All these speech filters, they were really quickly able to identify patients who did have Parkinson's and didn't have Parkinson's. And so this is kind of an amazing thing because it's basically using the latest signal processing. It's these guys, Dr. Saurabh Dasgupta, who's one of the world's experts on control and systems engineering, and his graduate student, Fahim Anjum, were able to do this very quickly. And so we're excited about this because we're wanting to see, does this work for other types of dementias? How early can we identify these patients? Does it predict future sequela? And does it do better than me? So I can tell you that if you come to see me, I think I'm an adequate neurologist. My statistical accuracy is around 85 to 90% diagnosed Parkinson's disease. And that's true for any movement disorders neurologist, because we have to make inferences based on how you move, what proteins are aggregating in your brain. And we can't look in your brain. So, we're not going to be 100% accurate. And you might think, well, it's 2019, we have those self-driving tractors. We have no blood test for Parkinson's. It's still clinical diagnosis. It's still clinical diagnosis for Alzheimer's disease. There's no blood test for Alzheimer's disease. There's no brain scan for Alzheimer's disease. There's supportive evidence, but it's still a clinical syndrome. So maybe we can get some objective data, and maybe that'll help diagnose. So we're excited about pushing this forward. All right, so the next thing I'm going to tell you about are a couple of mechanisms that are at work at Parkinson's. And because I think the Wicklunds have heard this before, I'm not going to tell you about optogenetics that Ted mentioned or neural recordings, which I was very happy that Ted showed you some neurophysiology data. I'll tell you about some other stuff that we're working on. So neurodegeneration is really about toxic proteins aggregating in neurons. And I was very happy that Peg showed you a neuron, so I don't have to go through it. One of these proteins is alpha-synuclein, and this is the way it looks natively. It's a it forms these beautiful structures, and it very complicated folding. But the problem with Parkinson's disease is this protein tends to fold out of the structure into these very complicated sort of schmutz. They're fibrillin-based beta sheets. They don't have the structure anymore. They sort of stick together, and they end up as a sort of huge intercellular balloon of alpha-synuclein and ubiquitin. And this is really bad because this leads to massive dysfunction in this neuron. We're not sure why. But if you look in the patient's brains with Parkinson's disease, you'll see this type of aggregate. You'll also see these aggregates in Alzheimer's disease and in other forms of dementia. And so we took a very simple approach, and this actually was a pilot project funded by the Carver Trust a long time ago. Hold on, let me just see why it's from. There we go. We designed a virus to use RNA interference, which is a very kind of fancy way to knock down proteins, and we were able to knock down the alpha-synuclein, and we found when we did this in our animal models, it was quite protective of some of the behavioral deficits. These are sort of measures of movement, how quickly animals fall. If you overexpress alpha-synuclein, animals fall, and if you protect against alpha-synuclein, they stay on this rotor rod, which is a spinning drum, longer. And so this is really exciting. We're currently stuck because we're sure it works in animal models, but we're not sure how to get it to the next stage. So if any of you have any ideas, I'd love to talk to you about how to sort of move this forward. We need to do it in primate models, and we need to think about doing it in some human trials, but we're a long ways because we have to establish safety. So the next thing I'm going to tell you is probably the most exciting thing I'm going to tell you today. So for some reason, Mike Welsh, I don't know if you know Mike, he's one of the best scientists here at Iowa. He's a pioneering sort of investigator in cystic fibrosis. He's really pioneered a lot of the physiology of the protein that's affected by cystic fibrosis, the CFTR receptor. So Mike was interested in energy. He's interested in energy in cells. And if you think about it, the one way to describe aging is that cells have less energy. They have less energy reserve. And you might think, why is that important? And if you want to see why that's important, in garbage collection, garbage being synuclein, if you stop paying city employees, one of the first things that's going to build up is garbage, right? And if synuclein, misfolded synuclein is garbage, you can see why it's important that a cell has enough energy. So Mike was interested in energy, and he was looking at the first step of the way cells generate energy. It's called glycolysis. And there's an enzyme called PGK, which is the first enzyme in the glycolytic pathway. And he asked the question, which is a question that I'm not sure why anyone hasn't asked before. How can we make PGK function better? And he found a compound that binds to PGK and makes glycolysis function better. And this leads to a whole bunch more free energy in cells. But what's amazing about this compound is Mike took compounds that are safe and effective and already prescribed. So there are patients who are taking this compound, thousands of patients. We have lots of safety data on these patients. So when Mike came to me, he said, well, could this work for Parkinson's? I said, I don't know, Mike. So Mike went and tried it in like 9 models of Parkinson's disease. Very convincing. And so he wanted to know, would this work in humans? And so one thing that we are able to take advantage of at Iowa is we are a data center for the Parkinson's Progression Markers Initiative trial. So we went and we pulled this data, And we looked. So let me just tell you, this is a complicated graph. I'm going to walk you through it. On the y-axis is the UPDRS, the Unified Parkinson's Disease Rating Scale. It's the best measure we have of motor progression. It's basically me looking at people, or movement disorders neurologists looking at people and trying to grade their disease, okay? Which is why we need better measures. So that's the measure of motor disability. And on the x-axis is time. And I'm focused on the blue and the green lines here first. You can see that over 12 months, over about a year, patients get worse. Okay? That's what you'd expect with Parkinson's. That's what I have to deal with every Tuesday morning. I have to tell patients, you're going to get worse. In this database, about 299 patients, 13 of them were on this drug. And I don't have to show you that this line is statistically flat. It's different than that line. And so this is really exciting because not only do we have data that this is we have a reason why it's protective, it increases energy. We have all these animal models showing that it's protective, but we actually have data that's protective in humans. So Mike and I have schemes to translate this and figure out how to study this at scale in Parkinson's patients all over the country. We're really excited about this as a measure. And we might be excited about the specific molecule, but I'm really excited about this as a mechanism because this is a new mechanism for neurodegenerative disease. Okay, so that's really exciting. But what's really exciting is, and I'm not going to bore you with this, but let me see if I can get this video to play. There we go. All right, so those axons, those dendrites that Peg showed you, here they are functioning in an awake, living, thinking mouse. So this is work by Georgina Aldridge, who I'm really excited about because she is going to start as faculty here at the University of Iowa. She trained in my lab. She's a neurologist. She's a specialist in dementia with Lewy bodies and cognitive disorders. And she sees patients and she does experiments. So the experiments that she does is she takes a very expensive microscope. She puts a small, precise hole in a mouse's head, and she looks for transients of calcium, which is an indicator of neural activity. And for me, this is like looking at, what you want to know, the stars at night, the most amazing things in the universe. This is a network of neurons, and you can see the calcium is flowing into a dendrite, the neuron fires. I'll show it to you again. And so we really are interested in understanding basic things with this measure. How does synuclein affect this network? How does taking away dopamine affect this network? How does amyloid, how do other proteins that are affected in degeneration and neurodegeneration affect this network? And so there's lots of really interesting science that Georgina can do. This measure enables her to look at neurons, blood vessels, microglia, whatever process you're interested in. And so I'm really excited to see where Georgina takes this. All right, so let me finally tell you about the stuff we work on most of the time in the lab is how to fix failing circuits. So, and this is a collaboration. One thing that's really unique at the University of Iowa, Aaron alluded to neurosurgery. This is Dr. Jeremy Greenlee, who is also part of our Neuromodulation Program of Excellence, funded by the Carver Trust. Jeremy is one of the best neurosurgeons I've ever worked with because you can talk to Jeremy about anything. If you have an idea, he'll be excited to figure out how to translate this. He's right now implanting deep brain stimulation electrodes in one of my patients with Parkinson's disease. You can see that this is, I compare it to better than a root canal. It's not pleasant, but our patient is awake, he's alert, he's functioning, and he's having these electrodes placed in his brain. But the payoff is here. This is not one of my patients, this is from YouTube, but I'm going to show you the effects of deep brain stimulation. So here he is with... Hold on, he's Australian. I'll turn off his volume because he's a ham. Well, we might be forced to listen to him anyway. So you can see he's got Parkinson's disease. He's got a really bad tremor. But by carefully mapping the anatomy of movement in primates and then rodents for 20 years, neurologists and neurosurgeons were able to figure out exactly where to deliver current to correct this problem. And I'm going to show you kind of what it looks like. Oops, let's go back. And here he is with the deep brain stimulator on. You can see that the statistics of movement have dramatically changed, and we're able to treat his symptoms quite well. It's a powerful it's not a cure, it's a powerful treatment. Now, but it's a paradigm shift that Aaron alluded to. You might think if I have a disease, I probably need some sort of pill. But he's got electrical current. And Ted told you that there's someone who's mapping the circuits of feeding. You might think about a treatment for obesity where we deliver targeted current. Peg Peg referred to addiction. There are circuits to control addiction, and we might be able to modulate those circuits. Aaron referred to depression. Certainly, we are manipulating those circuits. We might be able to deliver targeted electrical stimulation to failing circuits to correct them in real time. And so that's something that, 50 years from now, I really hope that that's something we're working towards. But it's going to take a lot of work. Francis Crick didn't get there. So one specific application that we're working to is in Parkinson's disease patients have cognitive symptoms. This is a very elementary cognitive task. Green is good. And with deep brain stimulation off, our Parkinson's patients don't do very well. With deep brain stimulation at 130 hertz for motor symptoms of Parkinson's, we found that they didn't do very well. But if we turn the stimulation to 4 hertz, this is a rhythm that patients are missing in Parkinson's disease, our patients get better. And I was surprised by this result, but I didn't really believe it until this group at Davis that I didn't know turned their stimulation to a similar 5 hertz. This is another cognitive task, and they were also able to observe an improvement. So we're now studying this again in 40 or 50 patients here at the University of Iowa to see if we can figure out how to correct not only the motor symptoms, but the cognitive symptoms of Parkinson's disease. You guys are from, you guys know the University of Iowa, but you probably don't know this. We're located here in this corner, right, in this building, in the PBDB, and the hospital is about 1,000 feet away. I know that because I go back and forth about 15 times a day. The main operating room is 6 minutes and 30 seconds at a quick clip. And, you can see that the reason I'm from Seattle, by the way, where Kurt Cobain and Chris Cornell were, which Peg referred to. But the reason I'm here at the University of Iowa, in Seattle, this whole complex is like spread out over the city. It takes 45 minutes to get across from the main OR to a research lab. Here it takes like 6 minutes, right? So this is an incredible confluence of medical and scientific resources. The VA, where we see a lot of Parkinson's patients here. My clinic is here. We see about 1,500 patients last year in our movement disorders division. So, we're able to merge this clinical operation with this research operation. This is unique, worldwide, doesn't exist. And so my dream is to leverage this and put a Center for Dementia and Cognitive Disorders here in the middle of this mix, where we can sort of touch base with all of these sort of operations and come up with new insights. And so I hope that we can work towards this. I think that this is going to be what's required to sort of bring back new therapies for patients with Parkinson's, new therapies for patients with Alzheimer's disease, and to really make a difference. And the last thing I might say is that I believe I bought some cards, so I'll hand out some cards. If you have any questions about neurology or anything in general, let us know. We're always happy to sort of field those questions. We want to be one of your trusted sources. Thanks very much.
Thank you, Kumar. I'm going to invite each of our speakers to come forward, and then we have the opportunity for the promised Q&A that I mentioned earlier.
Yeah. So the question is, and so you were taught well, by the way. So in discussing adolescence as a process in which you myelinate, is that still important in addition to the pruning? And absolutely, yes, it is. And so the two actually go together. So as you're pruning, you're increasing your white matter, which is myelin. And it happens in a specific way. It happens from the back of the brain to the front of the brain. So myelination and pruning and all of the maturational processes. So that's a good and a bad thing. It's a good thing that we have all that done. But the frontal lobe is the area of the brain that needs the most myelin. Those are the areas of the brain that have the most human functions. That's where social behavior is, attention, being able to sort things, being able to organize things. So you can see where those are the last to mature. So some of the, that's why adolescence is so prolonged, is because those are the things that take such a long time to mature. So it's myelin, which is white matter, and that, doesn't peak until about age 30, so.
Are is the panel aware of an article that was in Forbes that describes it's a vaccine that was developed to target the plaques using CRISPR, and they've shown in a mouse model, I think it was, that it actually reduced the plaques in the brains that were formed in those mice.
So the question was, there's a CRISPR-based CRISPR. I don't know if you've heard about this. Is one of the greatest revolutions in the last 10 years. It's basically using templating from proteins to target DNA, and so it enables you theoretically to edit DNA of humans. So there's several problems with it. You're born with, as you said, Peggy, all the neurons that you have even at the age of 90, right? So you need to get whatever CRISPR-based technology to all of those neurons. So that's hard. The second is you need to make sure that you're affecting a specific thing and not affect other things, because the templating isn't perfect. And so that's another major problem with CRISPR. But so this concept of decreasing the amount of proteins and plaques is exactly what we're going after. There are a couple of ways to go after. The one is this is a thing called antisense oligonucleotides, or I think that's going on for Huntington's right now, where there's little strands of DNA that target the DNA of interest. That's very exciting, and that's actually in clinical trials or moved. Yeah, it's in clinical trials and fairly advanced, not only for Huntington's, but for various genetic diseases. The second is RNA interference-based therapies. That's what I just suggested. CRISPR, I'd say, has these two big problems to deal with, and Ted can speak. Ted knows more about CRISPR than I do, but Ted can speak about those problems. But those problems really need to be addressed in mice prior to moving to human trials.
Yeah, I think a couple of comments. One, the work is, I think we have a sense of the targets for Alzheimer's disease. We know about the plaques A beta. There are also tangles, tau. One of the challenges is we don't know when to target them. And what you kind of heard from Peg could be summed up is that the child is the father of the woman. We give, it's a weird thing. We think of the woman being the mother, so the child is the mother of the woman. We think of the woman being the mother of the child, but actually what we have as adults when we're men and women is we have the derivatives of our childhood and our adolescence and our brain development over decades. And so the challenge with these neurodegenerative diseases and with other diseases is knowing when to intervene. With Huntington's disease, we have a genetic hook. We know the individuals that have the mutations that cause Huntington's disease. With Alzheimer's disease or schizophrenia or bipolar disorder or depression, there aren't single genes that drive it. They're polygenic diseases. So we don't know who's going to get Alzheimer's disease. And when you show up in the clinic, you might have been needed to be treated 10 years before you actually appear in the clinic. And so that's one of the challenges, is sort of reading the tea leaves of knowing how to go backwards. We're beginning to develop some compounds to image the plaques and the tangles, and that's helping us get back. And we're seeing those plaques and tangles when people are in their 40s, so 15, 20 years before they actually develop disease. And so that really is the challenge. One other thing we're finding, like you mentioned with vaccines, what happens in the brain is the result of what happens in the body. Right? So we have an immune system in the periphery that can regulate levels of A beta. We also have a metabolic system in the periphery, microbiome in our gut. And all of that can change how the brain functions. And so there's, for example, increasing evidence that metabolism really impacts Alzheimer's disease, and individuals with diabetes, for example, are prone to have greater cognitive deficits if they then develop Alzheimer's. So there's this interaction of the brain and the body. So that's one thing that is terrific to see somebody like Aaron Boes, who's in a large department of pediatrics that really is thinking about kids, but also focusing on the brain. So those are some of the challenges.
Other questions?
I've read by some writings about light therapy, and I forget the wavelengths, and if you comment on that, they had good results with mice, but haven't they gone beyond that?
I assume it's probably optogenetics. Does that sound familiar? Yeah, where you're firing lights inside the skull. Yeah. And it's a really effective way to modulate circuits in mice, probably the most effective that we have. How that translates into humans actually relates to a lot of the challenges that were just discussed. So in order for that technology to work, you need to change the genetics of the neurons that you want to alter, essentially. So you need to introduce new genes in a very targeted way, and then you need to shine lasers at it. I think it's a big, exciting question that it remains to be determined whether or not we'll get there in humans. there's certainly a lot of people working on it. That's very exciting. And how that competes with other technologies that are also in development, I think, is an open question.
Yes, Linda?
Yeah, this has been wonderful. My background is developmental psychology, and I can remember still in the late 50s when I had my first child psych class that the professor, Jean Burton, stood up and said, the first sentence, the trouble with child psychology is that we really don't have any. Really? So that's how far you've come. And along the way, you know, I became a developmental psychologist. You know, we saw Piaget, we saw what he was getting at in terms of how people develop. But also from my early college days, I remember one psych class where the professor gave us a book about the brain, and we all went, what? You know, it was just really like, oh my God, this was the future, he was saying. Of course, we didn't understand a word of it, and I understood a few of these words, which was interesting. So I've tried to keep up through my son, who's a neurodevelopmental pediatrician. And I also have an interest in the mind-body dichotomy that you study through ethics and philosophy and everything. And the question that is just jumping out at me all the time is, how much can we change people without changing who they are? You know, this mind-body thing, where do you draw the line? Look at the great people that you showed on the screen, and you wonder, is it necessary? I mean, I know it's not necessary for people to suffer. We don't want people to suffer. But the idea that these creative people had something wrong with their brains that you're going to fix is a little daunting. So I don't know, how do you have an ethicist on your team? Do you have a philosopher on your team, you know, to deal with all these things? Because what you're talking about is great for people that we know are suffering and pain. But what about?
So absolutely, you know, that ability and liability, you know, so that mental illness makes you vulnerable. The issue is, however, that we don't intend to fix anybody. And I'll give you a couple of good examples. So John Nash is a beautiful mind. When he came up with his game theory, which is what he won the Nobel Prize for, he was not sick. When he was sick, he said, I couldn't add a column of numbers. He passed on his math genius and his schizophrenia to his son, who had the same problems. Once again, when he was psychotic, he couldn't add up a column of numbers. When they were well, when their mental illness was treated, was when they were able to function. When you have addiction, you know, being intoxicated is not going to lead you to be creative. So typically, you know, being able to treat dysfunction allows that creative person to create. So treating mental illness is not going to diminish the creativity. It's going to allow them to be a functional person who can then produce the wonderful things that make our world so much better.
But Newton and Darwin didn't have these treatments.
That's true. That's true. Darwin's issue was he had panic disorder. So on the ship, he had a real hard time. But once again, you know, even though he might not have had treatments, in between his issues, he was able to function perfectly well.
So my wife's a philosopher, and we run over these mind-body problems with her department all the time. And so one thing I wanted to just convey is that when you meet a patient with Alzheimer's disease or Parkinson's disease, so with Parkinson's disease, they don't feel themselves. And when you can see when the patient turned his brain stimulation on, he said, you know, not with that patient, with other patients, like, I feel like me again. And so this neurodegenerative disease, it takes them away from And then if you've ever met a patient with Alzheimer's disease, you just talk to their families. Well, you talk to their families and you can say, that's not my grandma anymore. That's not my wife. That's not my husband. I can see them fading away. And so our effort as doctors is to bring them back to their best instantiation of humanity, to themselves. And that's our goal. And the last thing I might say is just to specifically, we actually do some of our stimulation experiments on normal mice. And I cannot make a normal mouse any better. And the reason is actually a very subtle thing about our brains, even though you feel like you might be better with more coffee, it's probably pretty close to optimal network function. And it's really hard to make an optimal network better. And that's not our interest. But when these both mental illnesses and neurodegenerative illnesses strike, it's incredibly painful for the people around to see this change in who they are. And so, if we can bring them back, if we can give people one conversation, that's my goal for my dementia, if one conversation with a loved one again to say how much I love you and how much that meant to, that's what we work for.
That's beautiful. Ted, would you like to make some closing remarks?
Yeah, I was just going to say, with the disorders we talked about, and when we think about our son Seamus with autism, he was in the neonatal intensive care when he was 4 days old at Children's Hospital in Philadelphia. And, there are kids with autism who have never spoken. There are kids with autism who have never gotten out of a corner and stopped stimulating themselves. There are kids with epilepsy that start having seizures when they're 3 days old, and they die from those seizures. So there are clearly dramatic impacts of these neurological and neurodevelopmental and psychiatric disorders. And those really need a cure. The child that's dying from epilepsy, the child that has never spoken, and you have no idea what their personality is. You're absolutely right that with disorders like schizophrenia and Alzheimer's disease, we have a sense of what the people are like, or with bipolar disorder, because in some cases they're fully functional, in other cases they're not. But there are many people with these particularly neurodevelopmental disorders, but also severe psychiatric disorders, that we have no idea what they're like. And I think one of our challenges is to try to find out what these children and what these adults are really like and what they're able to do. And to me, the whole view of the brain and the body as having many circuits, and circuits that mediate memory, circuits that mediate motor behavior, circuits that mediate emotion, means that we can think about particularly targeting those circuits that, for example, cause fear. But those are different than the circuits that enable creativity and memory. So this neural circuit-based idea of psychiatric and neurodevelopmental disorders, I think, is promising from this perspective of being able to target the symptoms that are most devastating, while leaving the characteristics of the individuals that we really find most creative and most needed in society.
Thank you. Please join with me in thanking Ted and Kumar and Peg and Aaron for their remarkable contributions, as well as their presentation this morning. I also want to take a moment to thank you for coming and to thank you for your support of the University of Iowa. As you all know, this is part of a series of a variety of opportunities that we are trying to share with those people who are our most generous friends and supporters of the University of Iowa. We welcome your feedback. We welcome your participation in our additional activities this coming spring. And at this time, I'd also like to ask my colleagues from the University of Iowa Center for Advancement to stand so we can say thank you to you as well. If you have any questions, any of us would be happy to respond. We do have an opportunity for some optional tours if you would like to see one of the labs. We have that lined up for you as well. So if you'd like to have the chance to ask a few more questions and maybe to dig in a little bit, we'd be pleased to have you do that. Thank you again for being with us this morning, and thank you, Ted and your colleagues. We appreciate it.